So our speaker tonight is Dimitar, and unfortunately I'm not going to try to pronounce his last name, I'm sorry, but he can do that for you. I don't like butchering things. I'm from Texas, and so I have that excuse that I can get out of pronouncing things correctly. So I'm in the cultivation section of the Mushroom Society, which might seem an odd person to introduce Dimitar, but since he's a taxonomist, all the other taxonomists in the society are so intimidated by this guy because he is so knowledgeable. He came out of nowhere a few years back and just started writing up papers and identifying mushrooms, and apparently didn't know too much to begin with and has just learned a vast amount in a very short brief time. So he's the author of quite a few new mushroom species in California and perhaps other places. So Dimitar is also doing things with genetic identification too, so he knows a lot about that. I saw him as a talk at Soma Camp a couple years ago, and I was so impressed by it that I wanted him to come and speak specifically about that topic. I believe that's going to be part of his talk tonight, but if he's not doing enough of it, I'm going to badger him to come back and do it again in more detail, just about the genetic part of it, because he explains things on a fundamental basic level where the average person can understand it, except that he has some people's always got too big of an accent, but most people say, no, he's like mycological enthusiasm on steroids. So he is, by the time you finish with his talk, it's like, oh my gosh, my brain is full. So, with no further ado, Dimitar will be our speaker this evening.
I definitely have to will have to step up to the task, so I'm taking my jacket off. Since some of you guys have been around and have seen my talks in the past, there will be parts that you recognize, that you have seen, so forgive me. I have had discussions in the general area of advanced mycology for a while, and I have done a lot of microscopy, I've done genetics, so here I'm going to try to stay a little bit more central without going into the excessively technical areas. I will just show you bits and pieces towards the end just to suggest how deep we can go if there are enthusiasts. The fundamental reason that I'm here is, as Ken said, I came in this field as an amateur about 9 years ago. I went to the Mycological Society of San Francisco Fair. I was dragged, somebody forced me to go to that place. When they told me that I'm going to see mushrooms, I was like, mushrooms? Why the hell do you need mushrooms? But once I reached there and I saw the diversity of material, I was smitten. That very next day I was in the forest, and I still haven't found my way out. So, this is, and I started like a lot of people just collecting and making photographs. All the colorful pictures here are just from one genus that actually became the genus of my study, which is the largest one. I do several other genera as well. So there should be a next button somewhere that I probably will be able to find. Okay, so let's start with a couple of general things, few pictures. Also, as far as the title of my talk, Professional Mycology for Amateurs, it's a little bit misleading because, let's face it, professional mycologists are disappearing. The field is becoming so narrow and they're paid so little that, they probably do it just for the love of the job. So that's what makes them amateurs. So I don't know if we're professionals or amateurs. I call it we're all amateurs who, some of us, achieve professional quality results. So the problem of fungal identification, you saw all these mushrooms, you see all these people walking around the tables tonight looking at mushrooms. What is this mushroom? What's that mushroom? This is the fundamental problem of how do we identify things. What features we look at, how are things related. It's another important subject because this allows us to group things and for easy understanding. And this is the problem that all of us try to solve at a different level. I kind of went a little bit deeper to really go, and to really be certain that the identifications are done to a scientific level rather than I think this is this or I think that is that. Most people, particularly those who collect for the table, their necessities are strictly limited. Number one is don't get poisoned, and number two, enjoy the hunt. I also enjoy the hunt myself. Most of my family, they're also purely hunters for the table. They make fun of me when I go and sit down and photograph some mushrooms for an hour. So, why do we care? Is fungal taxonomy and systematics useful? Actually, it is not. If you take a look at the industry, we are naming mushrooms or having a very solid understanding. It's turned out not to be a problem that will change, anything, or anybody is going to invest a lot of money in. This is the reason why mycologists and taxonomists are disappearing, and amateurs like me are really coming into the game. I wish to see more amateurs like me. I'm kind of like the sole amateur looking around who really goes into that particular taxonomy level in depth. But at the same time, it is science. It is important that we are able to categorize and understand the organisms around us and develop a better understanding. So, to me, it is very important, but on an industry level, it's not really. There are various journals out there who publish taxonomy and systematics, and just for those who don't know, systematics is the general science of organizing our understanding of certain organisms typically based on their heritage and how they relate to each other based on origin. So let's go off the boring side. So the number one area of practice that most of us do is macromorphology. Morphology fundamentally comes from the Greek word morph, which means form, is the study of the form, and most of the mycology that is being done at identification tables, forays, when we go out in the field, is macromorphology. We just look on the external appearance of a mushroom and try to define what is that. Here it is very important that I always try to suggest to people, even if they're rank amateurs, when you learn things, try to inject certain structure. Try to understand the general high-level groups before you go down deeper into the things and into the species level or subgenera. And here we have some basic groups which come across, like Ericales, these mushrooms that have gills. But then we also have the Rusulalis, which also have gills, but they're actually very unrelated, and we'll talk about that a little bit later with genetics. We have the Boletales, which typically, for the most part, have spongy hemiphoral layer. We have a lot of gastromycetes, this strange form of puffballs. Now, it's very important to know that just because certain organisms have evolved to these particular shapes, that doesn't necessarily mean that they are related. And I'll talk later on the genetic part. I always give it as an example. You look at the shark, the dolphin, and the human beings, and you'll normally think sharks and dolphins are far more related because they're in the ocean, they have fins, they have similar features. No, humans and dolphins are far more related genetically than sharks, which are completely different branch, much more action. So many times we discover that just looking at the external feature of whether they live underground, the Tachypuchias mushrooms, which are very common, they span all of the genera out there. Then we have the polypores. They're typically, for the most part, hard mushrooms, hard or semi-soft, and typically are saprophytes. So as I discuss these things, there are some typical morphological characters that we really pay attention to. This slide is a little bit old. I'll probably organize them slightly differently, not have it tagged or have it, is probably less important, but fundamentally we look at the gills, we look at the layers, at the area of the spores. We look also, very important feature, particularly with Agricalis, is the spore color. A lot of people learn to do this as a general organization, generally to organize. If it's a brown spore, then it's certainly in one group. If it's a white spore, it's something else. So for a long, long time, mycological research stuck pretty much on a macro level. The founders, of mycology, Linnaeus and the early 18th century guys, up to probably mid-19th century, including Fries, they did not use microscopes. They pretty much identified things at that level, and they established some ways of categorizing and established systematics, which later on authors who used micromorphology changed significantly. So to me, this is a good level if you're going to be collecting for edibles. This is a good level also if you're a kind of an interested in mushrooms, but not quite as to kind of go crazy like me, you know, into much deeper subjects. And here is a point that, you know, I started also by opening this, by creating this website where I have a great number of pictures of mushrooms, and that's how I started really learning myself. And I also used to, you know, record my trips. And here is something about the spring forays. I'm just digressing a little bit from my talk because you guys spoke about the spring forays. It's unbelievably beautiful out there. You know, it's blue skies, colorful mushrooms. There is nothing like spring in Sierra Nevada. It's totally enjoyable experience. So keep that in mind. So the next step that probably 5% of the people, of the amateurs, really go into is to utilize microscopy and the ability to look under the skin of the mushroom. You start discovering a lot of interesting cell structures, spores, and other features which are really critical. For example, these particular round spores, which turn blackish when they're treated with iodine solution, automatically separate all of the species, all of the genera, subgenera in the Drosselales group. You know, although they have gills, you know, sometimes, and even we had a couple of species here at the table, I said, you know what, if I just look at it for 1 second under the microscope, I'll tell you. We just typically look for major separating factors. These cells here that you see are very typical in the small mycenas. Small mycenas are sometimes very hard to identify using macromorphology, but once you look them under the hood, they give you all kinds of things. These tips of are typical for the Pezizas part of the ascomycetes. These kind of coloration with cotton blue, you know, kind of sometimes gives you a lot more information about the coral mushrooms and so forth. So I spent about 2 years of intensely collecting, drawing, and studying mushrooms just purely at a microscopic level. Here I have countless photographs of spores and what they, you know, and they're really critical aspect for identification. I kind of opened this thing with Firefox. Just 1 second. I should have opened it with Explorer because then it will give me a small thumbnails of all these different shapes. So we look at the spores, we look at other microscopic features as well. You know, the basidia. Usually the spores grow right on top of the basidia in the lamina layer. So there is a whole lot of microscopy. As I said, I have led and taught classes about 4 years in the summer camp, specifically dealing with that. One of the things that you discover that once you get into the microscopy is that you really need to open the literature. This is when the exercise of being an amateur becomes kind of more interesting but more challenging. And thank God we have enormous resources here in the Bay Area. If you just go to the UC Berkeley Life Sciences Library, you find some of the holy scriptures of mycology, going back to early 19th century. You have all these, the two shrifts, you have the French, you know, Journal de Société Mycologique de France, which goes from 1827. And you have the works of all of the writers. Some of these are precious, I mean, priceless editions, and, you know, they're out there. So as an amateur, you know, the sky is the limit as far as what we can achieve. So once you get into the microscopy, this becomes very, very important. So I'm just going to browse for a couple of photographs here. Sometimes you have to go even look, you know, into these incrustations on the hyphae, you know, to really look for features and to really try to understand what is what, or these particularly inflated cells, and this is an important feature, so-called clamp connection. So there are a couple of sites right now like mushroom observer, and there are probably about 10 amateurs who go really deeply into microscopy. And this is certainly a great way to get a lot more knowledgeable professional. But microscopy dominated the field of mycology from about the 1850s to the 1980s, early 1990s. And we realized that there are also a lot of things in microscopy which are pure, what I consider pure invention. People see a lot of things sometimes that don't exist. And I always give it as an example. Imagine that you have 5 corpses of people from different races, you know, and you just try to identify them and look at pieces of liver and a piece of heart. Now you can say that it's a human being versus not necessarily being something else, but between closely related species, you know, I don't think that microscopy is particularly helpful. And here is where going to the next level really became essential. It was very clear to me that I have to go one level above and really get into the genetics, the fungal genetics, and generally the utilization of genetics in botany became the norm in the mid-90s with some fundamental work done in Duke by several scientists, but Moncalvo and Pelcale stand out. And finding such so-called genetic markers that really allow you to classify organisms took some time, but right now we have arrived at the position that they exist. It is very important to know that if you take, let's say, the DNA of most people, 99.9% is the same because it controls our internal functions. It is, you know, it controls our heart, it controls everything. There's so many things going on in our body that are completely controlled by it. We can't change it. So you have to find these pieces of it that really are useful. Now, obviously, in crime scene investigations, they have found ways to point out that certain murderer was this guy and so forth. But then we need some sectors that can tell you this guy, you know, something that can tell us, hey, are humans more related to the chimpanzees or to the orangutans? And those are not easy to find, but they were found based on comparison of a lot of organisms. So here is one example, part of my studies, and I'll get onto there, are the yellow amanitas that we find. And we find a bunch of these, and we kind of had a feeling that they are falling to 3, 4, 5 species. But in all our conversations, we have bumped these. We call them either Amanita gemata, after the European species, or there is one that's kind of distinct, Pseudobraconii and so forth. But here is one particular example. You look at these things. I personally don't think that microscopy will help you here. In fact, there was a French mycologist, Marcel Bon. They made an experiment with him because he was a great microscopist. They gave him 5 pieces of the same mushroom. He came up with 5 different names and very convincing arguments. Like this spore is a little bit bigger, and I always give that as an example. You can say that the Norwegians are generally let's say an inch taller than the French statistically, but go on the airport and tell me like this guy is a French or this guy is a Norwegian based on that. Some things are statistically true, but they are not really useful in practical work. A lot of the spore measurements that I see, I was convinced, and now after having done genetics, it's more than convincing that we can't really talk about. So this is one example, and I'm studying this right now, and even Aurora sent me a picture of a mushroom yesterday. It had a little bit of salmon coloration, which is this species, and he's sharp-eyed enough to kind of, you know, separate him, and I showed him immediately a tree, which I'll show a couple of trees later, and we were able to identify. This one particularly actually is yellow, but this one is it's a very close sibling of Muscaria. So interesting stuff. So tell these apart. And here I always give this example of the brown quartz of Sierra Nevada. I published 5, 6 new species in these. But 3, 4 years ago, I go and dig these guys out, and I was going nuts. You know, you look at even the spores; they're quite similar, and you don't know, is this one species or several species? Some of them have this very distinct yellowish coloration here, which actually has fluorescence. But even that, we have 4 different species. So I used to collect material, and we, you know, fundamentally, I mean, the reason we're doing this thing is because we are sickly curious. I was just going, let's try to know what exactly these things. How do you tell these guys apart, one from another? And there are more, you know. This one is a very common one. You look at the spores, they're all elongated, and, you know, Ron Pastorino has been one of the great collectors of quartz, and he comes all the time with these guys and posts pictures on Martian Observer. And even then, you know, you can't even tell apart. You can't even tell them apart. Or let's get another example. For these, this is one group called anomalous. They are so similar. They have these tiny spores, which are all in the same range, and kind of similar coloration. Go tell them apart. These are all, by the way, different species. And, you know, this is when you realize that even very seasoned mycologists from the 70s and the 80s made, you know, gross mistakes, or they just didn't touch this genera. This genera that I work with on, probably the largest one, Copinarius, is generally considered untouchable. That's why I was challenged as an amateur. Now, looking back, I really didn't know what I'm jumping into. You know, I was brave enough to think like, yeah, nothing can defeat me, but yeah, this thing is really good. Or, you know, I always give an example, these yellow coats. You know, I was going for a while like that. These the same, you know. So now all these answers, I mean, I have the answers of all these questions using genetic means. This is one of the particular quartz in our area that we find quite much. And again, look, the similarity is such that you can spin a tail, you know, both ways. Are they the same or not the same, and so forth. And the 2 Amanita species that I published there, everybody has been knowing for a while, you know, what they are, but no microscopy could actually definitively say, are they the same as the European, or are they not the same as the European, or the spring cocora, is it the same as the fall or not? 4 major gurus in mycology. Here's Singer, you know, was this Austrian guy. Moser came, and Alexander Henschel-Smith looked at it, and they couldn't quite agree. So we took genetics, and the question was answered immediately. Understanding phylogeny also is important because, and I'm going to go a little bit faster here, it allows you to understand how generally mushrooms are grouped. We realize that the so-called ascomycetes, cup fungi, and so forth, they are really very different evolution than the general mushrooms that we have, you know, the bulids, the rosulines, and so forth, the basidiomycetes. And also very important feature of genetics is that it allows us to see which characters really are definitive and defining, which are definitive really to show us ancestral relationship. As I said, if you just look at the shark and the whale, just because they have fins, does that mean that they are more closely related than the human? It turns out not. It turns out that the structure of their bone structure is a lot more definitive to separate what a shark is versus a whale. Certainly, you know, the fact that they are mammals, it's a very definitive character. So once we start looking at the genetics, then it actually becomes easier when you go in the field. And it's easier for you to make sense of things. I'll actually have to go back to this slide in a second. Let me show you some practical implications here. For example, can you tell me which mushrooms are on the upper row? Which mushrooms are related? Or if you don't want to guess, for a long time we believed that the chanterelle and the gomphus were in the same group. Because they kind of have these outside grooves, you know, they look kind of physically similar, vastly not so. This guy actually is related to this guy, and they are more related to the Bulids. Actually, not to the Bulids, but they are in the same group, so-called Gonfales. So using genetics, we kind of realize that, just this external shape is not necessarily definitive. On this next row of mushrooms, these are two puffballs. But this puffball is closely related to the agaricus, and this puffball is related to the Bulids. That, there was a time where anything that was a gastromyces was considered like, a separate group which is genetically equivalent. Turns out not to be the case now. In truth, it has to be said that even in the 20th century, there were a number of mycologists who already understood that just because things look alike does not mean that they are related. So this is not as much new, but certainly some things in this particular group became known and apparent recently. And this also has an impact on, you notice that a lot of genetic names are changing. It's a complete flux. Because we have realized that a lot of things are just not what they look like. Even the Caprinus is a very good example. Just because some mushrooms turn into ink does not necessarily mean that they are closely related. These features have evolved, over years several times. So, and here is another case. These actually all four are genetically in the Antuloma group. Here genetics is okay, but because we know looking at the spores that these are closely related. But what was not known is that, for example, this is what we call Leptonia, and this is what we call Mulania. A lot of our mycologists, particularly in the United States, and David Largent is still active up in Humboldt, they believe them to be separate subgenera because they really, really look alike. I mean, I can tell Mulania from afar away, and I can say Leptonia with these beautiful colors. In fact, we had one of these down on the table, somebody wrote Antuloma correctly. It turns out, though, that when you do the genetics of Antuloma, these Leptonia pop up at various parts of the genetic tree. Therefore, they are not, here is a technical term, monophyletic. Monophyletic means to have a common origin. So therefore, just because they look kind of bluish and pretty and are very convenient to group as subgenus, that is not a true subgenus. It turns out to be rather a human concoction. And in these cases, it is really nice to know that stuff. And it wasn't definitive until we started doing genetics. Multiple arguments took place in the 80s between European and North American mycologists, namely Largent and Nordelus and so forth, about what is what. It was a very common thing to publish systematics. Every major mycologist of the world published his own version of the truth. Some of the most prominent were Singer, the French mycologist, the French mycology school, which is very strong, had a number of guys who did it, Moser, and everybody came a little bit differently. They grouped things a little bit differently. In fact, it's the book Modern Systematics from 1986 that Singer wrote. Today it's completely useless. It's totally useless because we already know about that. It's interesting to read it, but it became a archaic reading. It's more interesting to see. It's almost like looking at old movies trying to predict the world and how wrongly they got it. But we have to give credit that in some cases, the traditional mycologists were very much on the line. Other field implications. Here is a perfect example. A lot of people tell me, oh, now with this, molecular science, do I need to have a device when I go in the field? Mushroom hunting becomes less interesting. I actually think it becomes a lot more interesting because now I really know what I'm looking at. Now, the only reason I show these pictures is because you see these grisettes a lot in the field. And trying to make sense of them has been always tough because, again, looking at the microscopy here, worthless. All spores around. You can't really do anything. But we realize that with closely related species, you have a lot, if you're going to see change between them, that usually is manifested externally more than internally. And again, I give it example with the people in the races. You can see the different races by the color of the skin. You can tell them apart immediately. but you're not going to tell them apart if you look underneath, because the internal organs are a lot more conserved and don't evolve as much. So here now, when I look at the grisettes, and the genetics clearly proved it, they fall in three very clear groups. The colorful ones, that includes the Amanita crocea from Europe and our Amanita velosa, which is a grisette, it's a yellow, beautiful one. Then you have the gray ones, but the gray ones have one specific feature of the group Fulvi. They have this brownish staining, very intense brownish staining. So this is my way of really separating between some of our Amanitas like Amanita vaginata group and this one with the brownish stains, which are very prominent. And what was the name of our Amanita?
Pachycolia?
Yeah, Pachycolia, precise, thank you. So now, to me, telling Pachycolia from any of the other grisettes is a instant thing. Once I see this browning, now, Fierce got some, he wrote that both turn brownish, but no. You know, the vaginata group does not, these guys turn distinctly brownish, and you can really say Pachycolia. Because I've had arguments with one of our notorious amanitologists in the area about, oh, this is this, this is that. Arguments are gone now. Now you know some of these basic litmus tests. Another example, you know, I hunt for a lot of Russula. And I personally believe that this is the toughest group.
Russell is tougher than Cortinarius?
I actually am starting to think that Russell is tougher than Cortinarius. The problem, and you know, for the following reason, Russell is also a very large genus, and some of these, there are two main reasons why I find it harder. The coloration ranges are such that you cannot anymore just rely on one particular, you know, color, or it's green or it's red. You have to learn to read a range of colors, and they do have them, and that becomes much easier. This is a tree of something like 1,000 Russulas. Contains material from Europe, contains material from the United States. I have more than 450 sequences of local material. And you look at this tree, and it starts making a lot more sense when I plug pictures of it. And that's when you kind of start seeing that the range of the features. And the other reason why Russula is very hard is because, unlike Cortinarius, where nobody touched it before, people were scared of it. You know, only Alexander Henschel-Smith in the 30s published about 50 species from California, and then he was a very young mycologist. Very clearly, he didn't know what he's doing. He was already falling behind the European mycologists at the time. Now, this is a little bit of heresy when I say that, but he was clearly behind the European mycologists, so he didn't know what he was doing. And then in the next 8 years, nobody touched anything until I kind of parachuted down somewhere. You know. So, but the Russula, everybody has put its hands on it. And you know, it is such a genus that can make a fool out of the most experienced, you know, taxonomists. We found that, for example, Heritiers' sequences, you know, and mushrooms, they were not just mildly wrong, they were grossly wrong. They were just as wrong as, you know, as you just, toss coins. So, but it is very hard genus, and no matter how much I study, I still get punched in the face regularly, and I even sometimes get annoyed with myself because I believe that I'm at a stage already I should walk around with the power of knowledge, and every once in a while, not every once in a while, with Russula particularly, it's a common event, we sequence something and it's like somebody hit me in the face, like, you know, it shows me that it's something not what I thought it is. But for me, it has been a very practical thing to take these trees, see how things are related, see the big groups, and then you realize how some of them group into larger clades, and then let me show a nihest tree. So here now, when I build my trees, I start attaching pictures. And I start getting sense of the colorations, you know. There are certain ones where, you know, what is very important in Russula, we realize, is that color of the spore print. That is a master, identification thing. You find that if you have white spore, it's not like one is white and next neighbor is yellow and the next neighbor is white. No, the white spored ones are a very distinct group, and that really helps. What we find is that the yellow spore prints are a kind of stay higher in the tree. These are the most basal here. The blackening rossulas, in fact, and the compact and the russula brevites, they're very big. You find them everywhere. In fact, according to some scientists, they should be treated as lactalis. In fact, they're closer to the milk caps, although they don't produce milk. So you start finding for yourself, and now it helps me a lot in the field, that the blackening rossulas in these, compact and so forth, they are a very distinct plate. And then you start finding the what I call the greenish rossula. What's that? Can you read it? Cascadensis. So, yeah, Cascadensis is one of those rossulas up in Oregon, but let's make this thing a little bit shorter. So you start seeing that, yes, how the groups are. These are the most basal ones that are closer to Lactarius. They are all white spot in this group. They tend to be generally considered unpalatable, although the Polish and Russians eat them to eat everything. And then you start going a little bit higher in the tree and start seeing this beautiful group of these also white spot. One of the group are the smelly ones. In fact, one of the rossulas out there on the table tonight that I call them Monolis is in this group. They are very smelly. They typically are very hot on tasting. They're also white spot. And then you start finding into the most beautiful edible rossulas. These whole branches here of the tree, these are all white spot species. These are the greenish, beautiful, Vescus, Cyanozantha. And then the more you go up in the tree of the white ones, these have spore print, which is a kind of a pale whitish, very much whitish, but slightly off-white. But generally, these are considered pretty much white spot. So building these trees now, to me, it's much clearer how they really group. And for example, I will go in the field and I'll just see a green rossula. I will look for the color underneath of the spores. If it is off-white, I know it's something in the Cyanozantha. Cyanozantha group. If it is, if it is white, it is Cyanozantha. If it is off-white, then you're looking for Vesca, Caterpillar. And then we have some of these very distinct, very hard-bodied mushrooms, rossulas, like Mustelina and so forth. But the reason I'm saying this thing is that now when I see a rossula book, when they group species that mix white spores, yellow spores, and all these features, and they concentrate on another feature which is not really that critical and important, we can burn most of the books. So to me, the basic way to study rossula particularly is burn all the books, forget everything and start looking at the genetics. I'm going to publish something on this stuff. I promise myself. I'm just very busy professionally. And something like that, it will be a lot easier to take even into a fungal show. And many times, it's easier for me and satisfactory to say, this is a rossula in the Cyanozantha group, rather than having to go down and invent crazy species. So going up to that particular tree in-house that way, you find that things group very distinct. Emetica, for example, is one of those groups that is very clearly snow white spores, and the coloration is almost always either reddish or whitish or creamish in this range. So here we are dealing with ranges. This is the typical, and on top of this, using genetics recently, we discovered that this one, this one and this one are the same organisms. Not the same species, the same organism. The same organism can grow them either red or white. And it's not like identifying down to a species, but using police like DNA to say not a species of rossula, the same, but the very same mycelium can get them out red or white. And this is the common what we call rossula sylvicula and cremoricol. Both of the names are not really applicable because sylvicula is an East Coast name. But this is, just to give you some of the challenges and some of the great discoveries. In fact, I didn't believe it because although we have to give him credit. At one time, he wrote in his book, these two mushrooms appear the same except for the color of the cap. And he had a point. But we always find in white spores. Then, to me, the sanguinea and the rhodopus are another distinct group, kind of a clade, which almost, they are yellow spores, they're hot, peppery, and they always have a stem that has some kind of pinkishness. And you see that they kind of group together very nicely. Not always, I'm sorry? Californiensis, no, Californiensis. From what I remember, groups into the group of those that decolonals. These guys that turn blackish from this type. Oh. That is the whole this decolonals clade that has orangish and so on. I'm pretty sure, but I'll get back to you. I have sequences of it, I just don't recall right now. And we go up the tree, and this is a case where, to me now, it's much easier. Here is the Californias. Decolonals, see? This is Californias. And this is one feature that Norm is talking about, that they tend to turn grayish, blackish. And now we see that they actually group nicely into a clade. And that is a pretty good characteristic to really define a group. So for me, going and collecting Russula now is a lot more pleasurable experience. At the same time, I'm commonly invited to events where they ask me to put scientific names. And I know we don't have good scientific names. So this is the hardest thing. I can't lightly put your scientific name. This particularly happens with the Forest Service. They pay me money, like $40 an hour, to go and collect mushrooms. They want a scientific name. They don't accept that this is a certain section and a section finalist telling me, guys, I can't with my eyes. I know that these names that you have here, they're not real. And the way they have them is because in all these lawsuits between Rainbow Coalition and logging industry, there was a judge order that said, these mushrooms are protected. I want from now on for you guys to make a survey how many times you have found these mushrooms. But the list of mushrooms that the judge used was given by some mycologists who didn't know any better, and this list is just wrong. And I understand what it does, but now I know better. So the hard part on the other side of knowing more is that people ask you what is the name of this mushroom, and I say I know exactly what this mushroom is, but I know it doesn't have a name. People don't like that as an answer. So anything unclear here about trees and what we're trying to do with genetics? We're trying to understand the lineage, the general ancestry. Is that clear? Anything clear about that?
Eventually, you're going to have to use that to correlate back to some of the important things as the habitat, host, and what ecological services they do.
I really think that the habitat and the that is a very slippery slope. Things migrate unbelievably. We can see with the Boletus edulis, you see that below Mendocino, up to Mendocino, it is vastly with bishop pine. And then in the northern, it immediately switches to spruce. I think that the habitat, if we try to explore that, it's very hard to just. It's very hard to really create a neat, picture. Some things have a very clear habitat, but for the most part, and I don't know why, for example, in Bulgaria and most of Eastern Europe, the Boletus edulis is a spruce there. You will never find under pine. Never. It doesn't exist. Under pine you have Boletus pinovilus. In places of Italy, it grows with chestnut trees. Go figure. And it is pretty much the same species now. So once we start playing the habitat, it's really I find it a very tricky thing. So before we go into questions, I'm going to discuss just briefly my research. So as you mentioned, I study three groups intensely by financing my own sequencing and extraction of genetic information. Cotinerius is the big one. I have published so far 11 species. There are 3 phlegmaceans, and just published one with 3 Telemonia. 4 or 5 of these grown in Sierra Nevada. I will have to publish at least 60 more species. You know, I have a lot of work to add. There is a major paper with, that I'll have to write with John Mirati, you know, who is a co-mineralogist in the University of Washington. There are a number of joint projects with other people. In fact, 2 more species were described last week that I'm also co-author. They are from Europe. They're not particularly relevant here. So Cortinarius, where a lot more work is going to happen. Genus Amanita. Here again, these are very well-known species, but what was needed is somebody to do the real genetic part. And a lot of people tell me, oh, why did you do Francherii? Why did you name it Augusta? Well, describing Francherii, which is everybody knows, it's not the hard part, but the real hard part was to go and collect European Francherii, which turns out to be not to be a single species. So I spent about a year going personally to collect it and talking to friends in Italy and Spain, because there are 3 subvarieties in the real Amanita Francherii, and they all look quite differently. And I have to do a lot of background research, you know, to see, is our species related to any of them? Because we know they're different. So there is a, you know, it's not just describing one species. None of my papers, I just put a new species, you know, I think this is this. I have to put a whole tree and justification as to, I mean, very visibly why this thing is different. And that was the difficulty, not the California thing, but, you know, because the name Francherii is a European, so I have to go and do that kind of research. In this guide, for example, we have a situation that people have been arguing for quite a long time. Is this the same species as is the fall cockle and the spring cockle the same species? When I joined the mycological clubs in 2005, 2006, I started to ask that question. And again, 4 leading mycologists, Singer, Thiers, Moser, and Smith, knew this mushroom very well. And they just were not sure whether this is the same species or not. I mean, recently we kind of suspected it is, but I can't just go and write it up. So Professor Mike Davis, who has helped me a lot from UC Davis, in the early days he sequenced a bunch of my material for free. And he had actually done 4 gene analysis, and they are very distinct species. But then the question was, is what grows on the oaks on the coast the same as what grows in the Sierra Nevada, you know, or what grows on the Madron? Yes, I thought actually they're different species, but and here is the habitat, completely different habitat. You go here in the Sonoma County and you find this mushroom a lot. You know, I find it in the marine watershed in some place in Boston Mountains. You go into the Sierra Nevada, you find it. But one thing I can tell, this whole group of the Calitrota type of mushrooms, I think that they're fundamentally originated with oaks, because there is one thing. In the Sierra Nevada foothills, you find it almost always in the black oak. And even if it grows in the pines, which clearly it switches, you know, it never goes above 6,000 feet. You know, you don't find it up there with the lodge poles, you know, up in the high elevations. It's a kind of foothills and higher foothills. So, you know, but the genetic analysis was the real deeper thing that had to be done, and that's why, you know, I kind of did it because nobody did it before me. There were a couple of ornithologists who were very angry with me, who said, oh, you just came on the scene and you described it. Well, don't you guys have had 50 freaking years to do it. Now, there was a little bit of another situation. Here is that one of the very known and beloved mycologists from the East Coast wanted to put a name on this one. The reason actually I jumped to this one is I was not planning to work on this, but what happened is that I heard the rumor that he wants to call it Amanita ichthyrobalana. And when I heard the name, I was offended. I was also offended as a Californian now, that the East Coast mycologists have created a complete mess with California material. All of our stuff was pretty much described as East Coasters. Somebody sends the material and they stay over there on the West Coast. They've never seen it in the field and they just describe it and they create gigantic mess. There was a huge mess that we had, not to mention that we have to solve a huge nomenclatural mess. What is the right name of the fall species? Is it Trout or Calitroderma? And it's a very involved subject. So I felt a little bit offended and I said, enough, no more East Coasters guys writing our names, particularly giving ugly names to our mushrooms. And I just jumped and I just wrote a manuscript in a month. I got a lot of flack about it. Oh, you stole his research. I said, I haven't stolen any research. They were freaking doing it for 5 years. We have, by the way, Professor Davis had published his genetic information about 3 years ago. They just slept. They closed their eyes. They didn't even see it. They didn't even know what to do with it. So that's when I said, you know what, man, too late, baby. Time's up. So we rolled it up pretty quick. Russell is something that I have ambition with Professor Mike Davis. We sequenced a lot to write. No species have been described so far, but I think we need to, and probably the first one that we have to name is our Cyanozantha. We have to give it a new name. One of the things that I am kind of running into problems is coming up with good names. I like to give them names that really suggest the origin of the New World of California. So a lot of these particular mushrooms I gave Indian names or local names. This is Ciscali. Ciscali means good tree in a Pong language, that's how they call the tenor. This is social for tenor. This one I call the Athabascan, for the Athabascan tribes, because you find this in the conifers from Alaska down to Mexico, all on the west coast. This one I call Tuolonensis, Tabularlensis. This one is Coconarius poloni. This is strictly on the rocks. This is to commemorate our Indian people. This is Trochiensis. This is a very strange species. This one fruits in the great abundance of the Jeffrey pine in one particular location, close to Truckee. I have found it every year in vast amounts, and it has never been collected anywhere by anyone, by any class. You know, I've spoke to Ted Jordan in the spring class asking, didn't somebody bring this thing? No. And you know, this is another little mystery. And here you really need genetic analysis to see whether these things in the tree of other species, how are they related. This one, for example, this one, the Ciscali, was vastly related. You know, and here another important thing that has to be kept in mind. A lot of people say, oh yeah, it's easy with sequencing, you just take the sequences. But do you know what to do with them? That's the real question. And here is where, you know, kind of becomes, the field becomes a little bit less accessible, and me being a math guy in computer science, I really thrive in dealing with probabilistic analysis and, you know, being able to understand the methods. So for this guy, just to understand where this guy fits in the picture, I went and played with every single tool out there. I spent like 6 months, like the complete nerd, playing with all phylogenetic software to, you know, Bayesian analysis, you call it maximum likelihoods, everything, just to understand what this guy did to a satisfactory extent. Of course, somebody will say, but how do you know for sure? Well, in all cases, the tools produce conjectures, but sometimes these conjectures are much more close to reality, and ultimately, you know, if you see repeatability, if you triangulate between several different tools and what they produce, then you can say, you know what, this seems like the correct hypothesis. Certainly, it's vastly better than doing the old way. So I'm sure that there will be future generation of mycologists who will see errors. And then there is a whole group of, there is one particular species that we find a lot in the spring. We call it Cortinaria subalpinus. And everybody knows it. It's a very common one. And I wanted to write it down as a new species, but it has exactly the same genes as something called Cortinarius clandestinus, described by Kaufman from Oregon, and it's very common in the Pacific Northwest. So then when I was about to say, you know, although they have the same, you know, genetic marker, they're so clearly different in habitat, and so clearly different in time, season of fruiting, and so forth, that we can still do it. Well, that turned out to be the case in California. I went up in Oregon and Washington when I started finding color schemes that pretty much intertwined between the two, and I could see that it's not very clear anymore even are they different because they fruit in the spring or in the fall. And this is one case when I realized that with the current technology, I don't have the ability to make a scientific determination, so I will leave it to the next generation of people. Smarter guys will come with smarter tools. I certainly don't want to be like occasionally old-time mycologists were so eager to put a name on something and just write down on paper, you know, that you know, later mycologists look at that and say this is wrong. So even with these genetic tools, we realize that in some cases, if we didn't have the genetic tools, I would have much easier to bring them to separate species. But now when you look at it, they have the same genes, it's much harder. You're like, wow. So, you know, this thing cuts both ways, I'm trying to say. Many times you see that in some cases we just don't have the necessary methodologies to really know what we're doing. And this is the first part of my talk. I just want to suggest that there is a second part of my talk, which is a purely technical aspects of genetic research and what happens. And cool part, we're not going to go into that tonight, but that's when we already start getting into the real nitty-gritty detail. I wish I had somebody to show me that stuff when I was starting out, you know. So, but I'm glad to share it. About 3-4 years ago, there is a forum that I created about 6-7 years ago. But 3-4 years ago, I started sharing more molecular data with several amateurs. A couple of them published papers as co-authors. A couple of them already know pretty well how to get around to do some at least basic analysis on things. We have great resources out there. If you're really hot, you know, if you're really curious about things, we can go into the databases, and just want to show you about these brown Sierra snowbank cods. Here is the picture again. So once you get into the genetic area, then you get an extra pair of eyes. You see these letters, and you see that, oh, this one is not. You see actually that they are slightly different groups. In fact, I was not hoping to see that. I was hoping for one of my other tools to come with a nice coloration. Once you start looking into their, you know, marker signature, you start seeing the differences and really group them, and then you go back and try to make sense of them and see what is related to what. But now here, at least with the second pair of eyes, I have finally properly organized them. And now comes the next challenge. How do you describe this thing so that somebody can tell it apart in the field? Not easy, and then I can honestly tell you some things we just cannot tell apart in the field, and we have to live with it. That's, you know, or once you have a lot of experience, and you know, for example, this one I can always tell in the field because I've gotten used to it to the extent that it is fairly familiar. But saying always, I say 70% of the fruit bodies. If you bring me one underdeveloped fruit body, I won't be able to know for sure. Of course, I can BS you that I know, but frankly speaking, it's not satisfactory to myself. This one I can tell apart also because it has a very distinct basal area. This one I know very well. This will get a new name, close to something called described Cotonalis haxii. So again, one more thing, you know, why it is, to me as a taxonomist, having this second pair of eyes makes it a lot easier to go in the field and collect, and particularly right now, I know what I'm after. Another problem is that most of the mushrooms don't interest me anymore. I'm after very specific gems. So now when I see a table full of mushrooms, it's boring to me. I'm just looking like, I'm looking for the one that I found 4 years ago and I have a single copy of, and I have the genetic data, and I have my own personal rule. I don't describe things without minimum 3 collections. I don't want to repeat the mistakes of previous generation of mycologists to describe things on very limited data and come up and write descriptions that ultimately are uninterpretable. And the other thing, everything that I do is deeply photographed, and you can have it for free. I don't charge $300 a book, you know, like there have been cases. Describe something and try to make money out of it. I do it for the love of the art. So this is my first part of the talk. If you guys don't fall asleep, I can take it to the second part. But that usually is for more of a workshop setting where I get guys who really have read a little bit, and then we can go into the genetical, you know, stuff and play with it. Any questions? Are we on time? How are we doing on time?
We're doing fine.
Okay. So
Thank you, Dimitar.
I had a talk about 4 years ago. I've had actually several talks where I went so long that I started to see people massively falling asleep. So that's why I tried to pace myself this time. I had a conversation on Cortinarius about 4 years ago. They actually gave me a bad name because a certain individual always brings it up, where I had 3 professors in the room finally saying, Professor Bruns and a couple of other guys were saying, enough. Can we go home now, you know? That's what I realized. So this time I want to pace myself.
Well, it does seem like the mistakes in the past, as you say, have been made because they had a limited set So the potential is that there still could be some oversights there, even though
But-
you're using more sophisticated than the past.
But you know, I often hear this algorithm and say, well, I mean, this argument that's saying that, and I used to bring that argument, this argument myself. Oh, you're comparing so little, but we have played with a lot of markers, and we know which markers work well. So, we really know that the markers, you know, that give you good separation, let's say on a species level, are not necessarily as good if you have to relate larger groups. So people have become savvy enough that, you know, and I have experimented. I paid for utilizing 4 or 5 different markers, which are common in rhino and botany, on Cortinaris, to see which one gave you better resolution. And, you know, ITS is the best. There is another one called IGS that also gives similarly good resolution, but there is a problem. All the legacy data in the databanks is using ITS. So I think that where we are going to learn more is we see different species which naturally evolved because there was some geographical separation or habitat separation. We don't know yet how different they have to be before they can hybridize. And I always give example with the human beings. You see different races. If we were all French mycologists, we'll treat every different race as a different species. But in reality, they made perfectly well. So this is not clear to me. Just because we see a different, what we define as a different species from the United States and America, they probably can hybridize problem. Although that you can make this experiment very easily with saprophytic mushrooms, those that are easy to cultivate, and see if they can mix. In certain general like Saturella, Smith, and the guys used to do that. That's a very convincing argument. But with the mycorrhizal mushrooms, you know, you can't quite concoct this. So that's why things which are very closely related, I don't touch yet, because I don't think that we have the technology. But if I see in the same area, like it happens in some of the, like in up in the Mendocino area, 3 different closely related crops to grow, I know that they are not hybridizing because they're clearly separated there. They've been there probably for a lot longer than humans have been alive. So. Yes, Ken.
Yes. Can you give sort of a ballpark figure or an idea for the rest of us? How much time do you spend on your professional life and your mycological life? And then of your mycological life, how much do you spend doing, say, collecting and analysis and microscopical work and the genetic work? In other words, how much tangible energy and so forth is
How much sleep do you get?
Well, you know,
you understand my question?
no, I understand very well. The truth of the matter is that in the last year, because I've been professionally exceptionally busy, I have done very little mycological work. Down to the extent that people started to wonder and calling me like, hey, you know, what's happening? I also lost a little bit interest. I used to be very active on a forum called Mushroom Talk. I kind of lost interest in general discussions or the subjects that I really want to talk about. Most people are not interested. But to answer your questions, in the early days,
Go back about a year and answer that.
when I started, I was out almost every day because I didn't know where to search. I went to all the crazy places. Some of them were very bad places, but that's when you start finding things. That's, you know, so I keep a strict log with photographs of every single day when I have been out. Everything is collected and so forth. So for about 5-6 years, I used to collect and preserve things into my own herbarium. Everything that I see, I collect, I identify. So between 2000, okay, I started collecting right after the fair of 2004, but in January of 2005, I really became very interested. I came to the MSSF meeting in March of 2005. At that time, I already believed that I knew more than 90% of the people, because you know I remember that me and my son as well, he was 7 years old. We became so crazy. We bought all the books and read all the field guides. So I already knew more than most members because I was just curious. At the same time, I cannot remember the names of the fishes in my aquarium. So embarrassing that the person who comes to clean my aquarium, I have to always tell him like, can you remember what is the name of this fish? So I want to show that, you know, some people tell me I can't remember mushroom names because your mind is not into it. So just because I remember that, my wife tells me that I don't remember anything else, you know, which is probably true. But yes, to answer your question more concretely, early on I spent a lot of time walking the field, collecting herbarium. What I did is, I never do microscopy in the fall. I do microscopy in the summer where there are no mushrooms. You know, in the fall, your job is to collect like crazy and preserve. So for 5-6 years, I became a generalist. This is where all these pictures of mushroom cells and so forth. I used to have a job at the time that wasn't very taxing on my mind, so effectively my mind was really free to roam. On top of this, I'm a hyperactive personality. You can't hold me down. That's enough. Most people like to take a sleep. To me, sleep is like that. It's like, I wake up and feel it's very boring. So, then I started to realize that I go less and less in the field because my interest became more channelized. In fact, I'm bored now to go. I heard Kurt say I just drove to Sierra Nevada just to scout. And I'm like, oh, God, if I have to do that, I'm not trying to have him scout and tell me what's going on, So I don't scout anymore, which is bad on the outside, because you pretty much limit yourself to finding. But then I rely on great guys like Ron Pastorino, who is a master scout. In fact, he found a species that we're going to name it. He's going to get co-authored, because he deserves the credit. There is another guy, Noah Ziegel, found something very interesting, and his is the best collection. It's hard not to give people scientific credit. So with guys like this, he has this cord that only he knows where to find, or he goes there on some Wednesday where I have 5 meetings. So anyway, my going out has diminished, but there was a period when I really jumped to the genetics between 2009 and 2010, where I read intensely the subject. I mean, I really hit the books, and immediately the results came. Because initially this was okay, or 100 samples, this is 2-3K. Maybe not much, but it's a little offset. So, I got a little bit of money from Forest Service to, but that didn't even cover, but lately I'm spending a lot less time, and I've promised myself that this year I'll finish at least 1 or 2 books. But I think that it can be done because there are people who go every day in the forest. It's simply, it's simply an aptitude. Some people get high out of finding a psychedelic mushroom. Some people get high on finding a bolete. I get high of writing scientific papers. Particularly because in the early days when I was starting, some professors gave me a little bit of resistance. So I had to really, I kicked the door. I didn't just politely ask to enter. I kicked the door and then I asked like, hello,
We do want the academy back for the second half.
The second half, I can gladly do it.
Explain it in plain terms. We want to hear all about the academy.
As I told you on the phone, I don't think that in this audience, it's exact. We can talk right now after everybody's gone. Easy. We can spend 45 minutes if you want. Yeah. I just don't want to, don't want to get these guys into the.
We may have another venue for you. How's that?
Sure. I think that a more close venue where we'll say, this is going to be a little bit esoteric, and because there's a lot to be explained, which I think is going to be interesting to you. For example, you hear some claim about a mushroom or something. You can easily go on GenBank. You can do some easy analysis yourself. Hey, you can check the work of scientists. Now I go, at one point, when I was studying, I was going, and every phylogenetic tree that I see, I go and check with them. You will be surprised how loaded crap and errors they are. Oh my God, just because somebody told you, it's a scientific proof. There is no scientific proof. A lot of it, and we find that some of the early stuff in molecular stuff that was done, I'd say anything done before 2005, you can burn the books. Because most of the time it's done. And using wrong markers for the wrong things were proven. There are several examples where you clearly see that the scientists are using the wrong markers for the conclusion that they're trying to reach. And this I can discuss and give you examples. This, and then we'll talk about alignment.
Randall's going to be closing shortly.
Okay. We'll go about transitional rates. It turns out that in these 4 DNA, types that we have, they don't transition equally. Certain ones of them transition a lot more often to others. And here is the basic of statistical analysis. So that's why we built some trees using these methods. But again, I'm seeing people ready to go.
No, I have one quick question. Go back
K is unknown.
Okay.
K is either A or G or something.
Yeah, yeah.
There are a bunch of these characters which are probable. But if you take a look at on the sequence diagram, you can see sometimes. And that's why it's always good to have multiple sequences. There's a lot to be said about sequence quality.